1, 2, 3. That's it. We're
Mm-hmm.
live.
Mm-hmm. Yeah, awesome.
Yeah, it's good to see you.
So nice to see you, Tariq. It's been so
Yeah,
long.
yeah, it's been a long time. So since
Yeah.
I think the last time we met physically was like it was at the Broad, right?
Yes, probably at
Yeah,
the Broad, but like I I don't think was I a student already? Perhaps.
and
I just
no,
started.
yes,
No.
I think so. I think so.
Yeah.
Yeah,
And now
I
I've
think.
graduated and
Yes. Now you're
wow.
doctor. You're Doctor
So
Manakongtreecheep.
it's probably been almost 5 years that we've seen
Yeah.
each other in person.
Yes.
No, actually, you actually visited.
Oh yeah.
Maybe
Oh yeah. That's true.
we had
That's
dinner.
true.
Yes.
Yeah, we had dinner together.
Yeah,
Yeah,
that
it
was
was
fun.
nice. It was not long enough, but I think I stayed only 1 or 2 days in Boston. I
Yep.
didn't stay long.
Yep. Very cool.
Yeah, so it's super cool to see you again, especially that you have a nice you have great work that we can talk about,
Yeah.
especially yeah, we can talk about anything. We can talk about the single cell stuff. We can talk about we can also talk about the mass spec stuff. Like both of them I find really really interesting. I've worked on both, so I like both subjects. I really like your Pepyrus project. I think it's your last project, right?
Yeah. So I think most of my work now is so for sure my background is in single cell, and we worked a lot together in single cell sequencing technologies back in the Villani lab. But nowadays, I'm very much kind of back to basic, doing a lot of synthetic biology, um, engineering, protein design. But it turns out that this is a very good a mass spectrometry, I guess, is a very good use case for some, innovative synthetic biology techniques that can help. So somehow my work ended up being related to mass spec, which you actually have done a lot as well in your graduate school.
Previous life.
But yeah, I mean, I didn't have any experience in mass spec at all. It just kind of came to be because of the application just works out really.
Yeah. It's yeah. Honestly, I haven't done mass spec work in a long time, but I still remember. I still read the papers, so I can see the value of your work. So basically, Papyrus, the idea is that to create libraries using E. coli, which I think is a great idea because it's cheap. Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
Yeah, so in the mass spectrometry world, especially in our field, which is immunopeptidomics. So I work, even when I was doing single cell, I was doing a lot of immunology. And as I transitioned into my PhD, I got more focused on the therapeutics side of immunology. So targeted immunotherapy, personalized immunotherapy. And of course, I joined the lab of Kathy Wu, who is really the pioneer of the cancer vaccine field. So her work in 2016 with the Dana-Farber team was like the first clinical trial for personalized neoantigen cancer vaccine. Just to give everyone as well a kind of like a primer or just a summary of what it is essentially if all cancers, people say cancers are all different diseases and needs different types of treatment, which is true. But all cancer actually have one main similarity, which is they all are caused by some kind of mutations in your cell, in your DNA. As far as I know, every cancer has a mutation in the DNA that causes, transformation in the cell. The cell becomes what we call malignant. And the way it becomes malignant is different across different cancer types, of course, but you could see that they all start off with mutations in the DNA. And this mutation actually can potentially create mutated proteins. DNA becomes RNA, becomes protein, the central dogma. And these mutated proteins, through the cell, mechanism, will always recycle. And the cool thing it does, and every cell in your body does, is essentially, other than red blood cells, is they will present pieces of your proteins that you produce in the cell on the surface. And this is called MHC molecules that do this presentation. So these proteins get degraded, chopped up into tiny pieces, and then these pieces we call peptides, which will say a lot of this word, peptides. That's essentially my PhD. But these peptides, once they get chopped up, gets presented on the MHC molecule. And, it's been a huge mystery and huge quest in the field of immunology to figure out how these peptides that get presented are recognized by your immune system. Eventually in the 80s, people figured out that T cells that we all have in our body have these unique receptor that can actually identify, millions of combinations of peptide sequences presented by the MHC molecules. And so, yeah, that was the holy grail of immunology. Like, how does this thing work? How do cells present peptides? And then how do these peptides get recognized? Or sometimes, we present all sorts of stuff in our body. Like, why doesn't your immune system recognize everything? Whereas, when infections occur, and your cells get infected, pieces of your virus can be presented, and then, of course, your T cells can then target those. And then this whole field of vaccine actually, started way before all this mechanism was figured out. But once the mechanism was figured out, we're now like doing a lot more in terms of like more precision vaccines. And to loop it back to cancer, I guess this is where our lab's work came from, which is this idea that if these cancer cells, they can mutate and produce all these mutated proteins, these proteins theoretically should be able to be presented, and the mutation should be recognized by these T cells as something foreign. Not every cell in our body has these mutations, only cancer cells does. So in a way, these T cells actually can treat these cancer cells like like an infected cells, like viruses, like bacteria, something foreign that's pathogenic and invading our body. So yeah, so that's what my PI, my professor, Dr. Kathy Wu at Dana-Farber, she came up with this idea of, let's sequence the mutations in every patient cancer cells. Patient comes in, you get your cancer surgically removed, and of course, vast majority of patients, the cancer will come back. That's just the unfortunate reality of cancer and why it's so hard to treat. But the surgically removed tumors, you can sequence them. And so, this idea came about that if you sequence all these tumor deeply enough, you get all the mutation that's in the tumor. You get all the sequence DNA mutations. And from that, you can design a vaccine or peptide sequences that contain the mutations. And then these mutations potentially could be immunogenic. So if you give this vaccine back into the patient, you're actually helping train the patient's T cells to recognize these peptides in the same way that the vaccines for influenza or vaccines for COVID help your immune cells recognize these pathogenic sequences. Now your T cells, with the help of the cancer vaccine, can actually recognize these mutated sites on the cancer cells. And then, you would know more than me, actually, since you do a lot of single cell work to understand how T cells, clone, expand, and what are their characteristics. But a lot of these T cells can then, become trained and become memory T cells or become effector T cells. And then now they can be activated and target cancer cells. So yeah, that's the whole journey of my PI and how she, in a way, built this lab and built this field alongside many other scientists, of course. But it's very unfortunate that I'm able to work in her lab, who is, one of the pioneers of this field of immunotherapy.
Yeah, it's it's great when I hear you talk about your PhD project. It's your PhD basically looks so much like mine.
But yeah, like this peptide and MHC molecules
Yes.
and the T
Yeah.
cells, and how do we find the right peptide MHCs? Of course, you did exactly that with
Exactly that.
a
Exactly
lot
that.
of predictive models, which is really beautiful work. And I mean, you should introduce us to this idea, actually, because I think it's not a lot of people know that this is one of the use case of AI and of machine learning, that you can build these predictive models that are directly can be used in clinic.
Yes. Yeah.
And I'm not on that side, but you definitely were very involved.
Yeah, but I was more involved. I was involved on the same side as you, but on the computational side, like more computational. So it started the same way. So we need to identify what we call these peptides. We call them like tumor-associated antigens. So how TAAs. So how do you get your TAAs? You need it. You have to go through the mass spec. You have to go through the mass spectrometry. And but the limiting factor is actually what do you call the database of the mass spectrometry. So what I did during my PhD was to do something very similar. Let's take the patients, the people, the subjects, sequence their genome, and then find the mutations, and then put that inside the database, and see if we can identify these peptides better. So that was like my side of the work was purely computational, like dealing with the combinatorial problems of having different mutations and things like that, finding a way that you can fit the database inside the mass spec. And it did work. It did work back then. And what you did, which I find super interesting, is that you did that, but from the biochemistry perspective.
Yes. Yeah. So, I mean, as you said, to find these peptides, you need to go through mass spec. That's the only direct way to find these peptides. You can take the tumor cells. Of course, you can predict these mutations. What do they create? Go through like the DNA will become RNA, will become the peptide sequence. But to actually find them on the surface of tumor cells, meaning that they actually get presented by the MHC molecules, you do need to perform mass spectrometry on the samples, and that's a really tough thing. And then from the mass spectrometry, extremely complicated spectral data sets. It's just like a bunch of lines that looks like a mess. To actually like convert that into a sequence of peptides, first you need a database of the mutated sequence, which you have done extensive work on. But I think what people have come to realize is that, in clinical samples, you get very few cells in the end. And of course, you can grow cell lines to, 500 million cells, a billion cells. But actually, in these tumor samples from the patient, sometimes you only have like 100,000 or a million cells. And that, when you translate that to peptide numbers on the surface, is very few. And I mean, as long as we find something, it's very clinically relevant. That's your candidate for a vaccine. So it's really important to find at least just one evidence of it. But once there's so few, and the detection has a sensitivity limit, it becomes very difficult. So yeah, just to get into our idea of the chemical approach. We basically just went one step further. Like you have your databases of the sequences, and they definitely help if you have a database of mutated sequence to try to match that database and the spectra that the database can predict to the actual spectra in your sample. But it's not perfect, so we thought, instead of using just the database, why don't we just generate all these spectra from the actual peptide? And we need to generate in large amounts so that we ensure that these peptide references are very high quality, and that once you map those mutated peptide sequence that we generate into the actual sample, if it matches with very high confidence. And people do that, but peptide synthesis is extremely expensive. It costs like $200 for peptides. Or if you want a high quality, high quality purity, the way people do it is they just drop individual amino acid onto a piece of nitrocellulose paper, and it binds, and then you wash everything off, and then you drop another. It's like a robotic system, but it's extremely expensive. It uses very specialized instruments. It's also takes a while to get to, and especially if you want to look for, among these thousands of possible peptides, which one are in our patient, you can't really make 1,000 peptide spectra from synthetic peptides. But our idea is that, the E. coli it can produce as many peptides as we want. And actually, when I was an undergrad, that was kind of a lot of my work was working with these E. coli, these tiny bacteria. And people over the course of, the past 100 years have utilized this bacteria for making all sorts of proteins in a very programmable way. So you can design this protein's DNA sequence to produce any types of peptide. You can insert it into those DNA into the bacteria at very high efficiency. You can grow it in like a vat, or you can grow it in a huge flask so that it keeps producing. So it's a very low cost and scalable model organism in a way for producing proteins. So yeah, the idea is very simple. Once we get the mutated sequence of the patient DNA, we generate, all the possible candidate peptides. So peptides we think we could find as a target that's presented by the cancer cells. And from there, we generate for these patients, tens of thousands of peptides, which we can do with easily in the E. coli. And then from there, we generate a spectral library. And this process is what I call Pepyrus, which is just a cute name.
It's a good name.
Pepyrus with a E, so peptide. A lot of people who do peptide work, they use these fun names. Like there's like a peptide company that produces synthetic peptide called Pepper. And of course, there's a very famous, not famous, but like a very big company called Pepprotec, which produces a lot of these synthetic peptides. So yeah, just a lot of pep names.
Yes.
Yeah. This one is kind of like a library of spectra. So that's why I call it Pepyrus.
Yeah. I think it's a good name. I think it's a very good name. It's also a very catchy name, easy to remember.
It's easy to remember. It's easy when we are in lab meetings and we have to distinguish like these are synthetic peptides, these are not synthetic, because we're benchmarking and all that. So it gets very confusing if we don't come up with a name.
Yeah.
Yeah,
that's true. There's something I feel like it's super interesting. It's also funny because it's exactly what I did computationally. It's like finding all these different peptides and putting them into the mass spec. But there's a big difference between what you did and what I did. Is what you referred to at the beginning. It's what I did was mostly in a research context. So we took the team. I didn't do it personally because it's a huge endeavor. It's like it's not just like one person cannot do all that. But basically, blood extraction, and then from the blood you create cell lines, and then you cultivate these cells until you have enough cells, and then you get them into the mass spec, and then you can do your identification using the database that I made. This method has several problems if you want to apply it clinically. The first one is it takes a long time to cultivate all these cells. It can take months. Second, the other thing is that you have to do it. You can do it on solid tumors. So that's also a big issue. So it's not ready. It's not a method that you can apply readily clinically. You can maybe use it to identify some TAA or tumor-associated antigens that you can maybe use for a vaccine. But if you want to do something that's really personalized for a patient, it becomes really, really hard to pull off. But with your method, it's actually doable. Yeah, which that's super cool.
Yeah, the E. coli grows very fast. It only takes one night for it to start producing enough protein for you to get it out and synthesize. So and each E. coli batch, each E. coli culture, you can make tens of thousands of peptides, unique peptide sequences. And from there, once you get the peptides out and put it into mass spec, the important part is the spectra of these peptides, right? They're references. They're reference spectra that you can use to map to patient samples. So an example I gave was by using the patient mutation signature, you generate a patient-specific library. But I think what is really cool is that we could also generate libraries of common tumor peptides. So these cancer cells, beyond mutating, they also become very different from your healthy cells, almost unrecognizable from your healthy cells. Some cells get bigger, some cells just expand, become different cell type. Some cells, they have this process called dedifferentiation, where they become more stem-like, more closer to what a stem cell would look like. People have seen this in like melanoma, which is skin cancer, but then they become more like neurocrest stem cell-like properties. And that, in a way, this type of transformation, in a way, is how cancer cells metastasize. So when people see how it, like, why does my skin cancer now is, you can see it in your lymph nodes, in your lung, and all that. Sometimes even in your brain, it's because they transform and become a unique cancer cells. And this is through a process. Of course, if cells transform, they need to produce new sets of proteins, right? You can't just become a different cell without producing new things. And these proteins, of course, could be presented as well. And if it's a process that starts from cancer cells, potentially those can be target that is cancer specific. So these cancer specific proteins, you mentioned it actually already, tumor-associated antigens, could also be targets for vaccines, can be target for CAR T cells. So another thing we are building is a off-the-shelf, spectral library. So we're generating these huge peptide data sets. We have, like, at this point, almost maybe 100,000 different peptides now, from 20, 30 different tumor-associated proteins. And potentially these peptides, when you get a patient sample, these peptide spectra can just be used as a reference, and you can map it onto patient sample mass spec data. And this is very rapid because the spectral data set, the files are already in the computer. You can use it to map to any new patient sample, and that you can get like a high confident, high quality mapping, which you cannot if you just do a normal peptide search. And there's some technical details about acquisition methods. We're also using a newer acquisition method where we actually fragment multiple peptide at the same time. And then we rely on this reference spectra to deconvolute the co-fragmented spectra. This is called DIA, if you want to search.
Yeah.
But that would get too technical, but it's a big part of our innovation. And it's of course, all up to the broad proteomics group who came up with a way to use these two technologies together.
Yeah, I think we can touch upon the DIA, like data independent acquisition.
It's a really weird name, and I'm not a mass spec person, so I would say I'm a biochemist and an immunologist. And I definitely do mostly wet lab at the moment. But yeah, so DIA or data independent acquisition, unlike what people have been doing for the past few decades, called DDA. So in DDA, you have two steps in a tandem mass spec. So MS1 and MS2. In MS1, once you ionize your peptides, you then, as it goes through the analyzer, only certain peptides get picked for further fragmentation. So you do lose peptides as it goes through the machine, through the mass analyzer, and also through the tunnel, these electric field tunnels. And so these, in the DDA method, as each peptides go through, you only picking certain peptides, mostly based on the intensity. So how abundant these peptides are, so that would lead to sensitivity issues, because not every peptide gets picked, not every peptide gets sequenced. But in DIA, the way they're trying to overcome this issue is that instead of picking one peptide at a time, you're picking a range of peptide properties called mass to charge ratio. So you're picking a window of mass to charge ratio, which would cover multiple different peptides of different characteristics. And then you just co-fragmenting them at the same time. You're just basically shooting them with with ions and then just fragmenting them all. And from those peptide fragments that's also ionized, you can then start to read the sequences of the peptides into its amino acid components. But because they're co-fragmented, it's just a mixed spectra of multiple peptides. And from there, you then need a reference spectra, like a clean reference. And this is where, computational models, they have their limitation. Of course, people are building really, really good models to try to deconvolute this. And I'm sure it's going to be, in the end, computational models are going to be the way to go. But for the moment, we are able to produce high quality real spectra for low cost at a large scale. So that's what we're going with at the moment. And we've shown that we can use this to map onto these and deconvolute the peptides.
Yes. So because you're basically tackling there's a huge issue if we do for that has been plaguing basically immunotherapy and electrification of these peptides is that there's two things actually. First one is that they're very small, so it makes the identification harder. That's the first thing. The second thing is the abundance. Right. They can, and this is actually a huge issue because if you think of a cancer cell is mostly a normal cell. It's still a cell from the same body.
Right. Exactly.
So the peptides that the cell representing are mostly normal peptides.
Exactly. Yeah.
Yes.
More than 90% probably are just what peptides you would see in any normal cells.
Exactly. That makes it a big issue because you want to identify like this small, like this very minute differences in peptide presentation. So what that means is that the peptides that you're actually interested in are not that abundant. And exactly. So if you do data like DDA, data dependent acquisition, you might lose these peptides. If you don't have the right libraries, you might lose these peptides also. So what's cool about Pepyrus is that you're basically solving these two issues at the same time. So you're creating arbitrary abundant number of these peptides. You can have as much as you want, and at the same time you're using DIA method to basically get the sequence of these peptides, which is also it's like. The reason why he said it, but it's not super easy to do because if you have one spectra associated to one peptide, it's way easier to identify the right sequence. But if you have a spectra that's many peptides and then you have to deconvolute them, it makes it way harder. So exactly. So I think also this is one of the coolest contributions of of Pepyrus is that you're really solving these two issues at the same time: the issues of abundance and the issue of identification. Yeah.
No, you put it perfectly. you understand the paper better than me, actually. No, that's exactly what we're trying to go for. And the hope is that, as we build this, we are also building the method that's easy for people to use. But at the same time, once we, like I mentioned, once we put these peptides and we create a reference spectral library, that's a file that anyone can access, really. So the spectral library, as we build it out, we can use it for multiple samples at many different contexts. And one cool thing we did was, we built a endogenous retrovirus library. So endogenous retroviruses, these are actually kind of part of us. We call them viruses, but it's because, like our cells and us as organisms have been fighting viruses for millions of years, possibly hundreds of millions of years. And these viruses, the ability to infect your cell and then, transfer a DNA into a cell means that sometimes when it integrates into your cell, it stays there as pieces of DNA. And, through millions of years, of course, these DNAs become dormant. They become so mutated across the path of evolution, but they're still there. We still have pieces of, hundreds of viruses that has infected our ancestral species since the dawn of time. And interestingly enough, these sequences, they can be translated. They can be, turn from DNA to RNA into actual proteins in very weird circumstances. And of course, cancer is one of those weird circumstances. And it's an emergent field that people are seeing through, very deep sequencing, through very innovative approach to map sequences that in cancer cells, sometimes you get RNA sequence of these viruses. And more recently, we're able to see peptide sequences of these viruses being presented on the cancer cell surface. So, cancer does weird things because it's just rapidly mutating, evolving, trying to fight off our immune cells. There's things like, broad hypomethylation, DNA hypomethylation, meaning that, your epigenetic, your methylation in your DNA is just all whack up, and things are just being converted to RNA like crazy. Your different transcription factors, things that are controlling and regulating which gene you're expressing in what cell types, are also like all out of whack, and they're just expressing all sorts of things that comes from our DNA, including these viruses. But long story short, we decided to build these endogenous retrovirus library. It's a very interesting area, and because we work with renal cell carcinoma patients, so we have a trial paper just came out a couple months ago from our lab that showed a personalized cancer vaccine trial for renal cell carcinoma. And then a follow-up paper from that came out basically back to back, explores endogenous retroviruses that are expressed in renal cell carcinoma. They're actually, quite highly expressed because of certain specific genes that are mutated in renal cell carcinoma that leads to this hypoxia inducible factor overexpression. And this is actually our work with Bill Kaelin, who's the Nobel Prize winner for the discovery of hypoxia and hypoxia inducible factors, how cells respond to oxygen. So this was a work from our lab with Professor Bill Kaelin. We have identified many endogenous retroviruses that are responsive to this hypoxic conditions and also to this specific mutation in renal cell carcinoma. So we designed a ERV library intended to be used for renal cell carcinoma patients. And we did find a lot of ERVs in these presented, these are so actually presented and potential cancer targets in the surface of renal cell carcinoma samples. But interestingly, during lab meeting, basically one person in our lab who does melanoma, so he's involved in melanoma cancer vaccine trial. He was like, "Can you search in some of my samples, some of my melanoma samples? What if we have ERVs? You never know. Like melanoma is highly mutated, and can transform into all sorts of very weird cell morphologies. I mentioned, I think earlier, that melanoma is one of the cell type that can de-differentiate and become more stem-like. So yeah, we decide to use our reference spectra, like these Pepyrus spectra for ERVs, designed for RCC on melanoma samples, and we did find, I think we did find 2 ERVs. They have not been characterized to be expressed and presented in melanoma, so it's very interesting. And because we actually, mapping real peptide spectra to the sample, we can be more confident that these are indeed, actual peptides that we're seeing. And yeah, so it's something we're going to look into. Of course, could be false flags, or we there's a lot more to uncover, but it's just showing the utility that we can very quickly search samples that has already been run. This was run actually last year, this melanoma sample, but we like, let's just search it, because the reference library is there, the sample data set is there in the mass spec. So yeah, it only takes 1 hour, 2 hours to to do a new search, and it's very, I think it can be very powerful.
It's cool. There's ERVs are probably like some of the most promising targets for cancer, because we've been focusing, the field has been focusing a lot on mutations, because they study, cancer mutates, but if you have, for example, for melanoma, like melanoma has a lot of mutations, but so do normal skin cells. They also have a lot of mutations.
Yep.
And
constantly exposed.
Yes, constantly exposed. Yeah. Exactly, constantly exposed to the elements.
Yeah. Right.
It's not just UV; it's everything. Like you touch stuff. There's a lot of things happening. ERVs are actually pretty interesting, because what cancer cells do is that they also play with the genes. So you can have a gene that gets translocated, like moved around, copy pasted. You can have the promoter, which is the part that decides if the gene is going to be expressed or not, that moves here and copy pasted to something else. And then what you get is not, it's a form of mutation. It's the DNA that's changing, but what's after it will get actually expressed. And that part, it could be an ERV. So if you look at, for example, if you look at cancer cells and the different numbers, copy number variations that you have in genes, you see that this is a feature of pretty much all cancers. And and what that suggests is that you probably have a lot of regions of the genome that not normally expressed, that are actually expressed in cancer cells, even though they're not mutated. Like this part don't have to be mutated, but because of mutations happening around them, they get presented, and these are probably like the most, in my opinion, they are part of the most promising tumor-associated antigens that we can use for a cancer vaccine.
No, for sure. And the beauty of it is that, especially in the case of the ERVs for renal cell carcinoma, these are driven by some very common drivers of the actual renal cell carcinoma malignancy. So, this type of cancer it's a kidney cancer, but it has certain characteristics, and we found that these characteristics come from common mutations that occur in these kidney cells. In this case, it's from a gene called VHL, which is a ubiquitin ligase. It's responsible for degrading proteins. So once it becomes mutated, proteins that should be degraded are not getting degraded, and then things go wrong from there. One of the things that happens though is that it starts to translate a lot of these ERV genes, and these ERV genes then starts to get translated into peptides and presented. So I'm not involved in this study, but study in our lab has shown, our lab and Bill Kalin's lab has shown that these ERVs, they can be shared among patients. So many different patients that have this specific renal cell carcinoma, kidney cancer, they can present the same species of ERV. You could call them, I guess, viruses are not living organisms, but I guess you can call them like a species of or a type of ERV can be presented in many different patients. And so this is a really highly promising target. And the cancer world is always trying to find a target that is specific for the cancer, because we all want to move away from chemotherapy, essentially putting toxic chemicals in our body and seeing if the cancer dies first or we die first. The field has always been trying to move away from that concept and trying to be more targeted towards cancer. So it's finding cancer target that's specific for cancer cells, but at the same time, it's not from this patient-specific mutation, but comes from something downstream of how this type of cancer works, or types of how multiple types of cancer works, and whether they produce this common antigen target. I think is a very one of the most promising area of research in cancer, in my opinion.
Yes.
If
definitely.
we could find something like that, it's what people call the magic bullet, which I think, funnily enough, was a term coined by this German immunologist / cancer biologist, I think 100 years ago, and he is the inventor of chemotherapy. But his ambition was to find this magic bullet, something specific to the cancer. He ended up finding something that's definitely not specific for the cancer, but it's still very good. Chemotherapy has saved many millions of lives. But the search still continues for the magic bullet.
Yes, the magic bullet. There are probably like several magic bullets depending on the cancer.
Yeah, it's gonna be a little too tough and a little bit too idealistic to find that. One thing that is specific for all cancer and not our healthy cells, but several cancer-specific targets that leave our cell as as unharmed as possible is the constant goal of this field.
Yes, definitely. But there's been some. I mean, the most recent developments are CAR T cells.
Mm-hmm. Yeah.
And before that, we had the immunotherapy with checkpoint inhibitors, yeah, which also can have some serious adverse events.
Mm-hmm. As we know very well.
Yes.
That's how we got to know each other.
Exactly. That's
from
how
this.
Yeah. And I'm still working on that. I'm still working on the topic.
Yes.
Yeah. Because the idea is immune checkpoint inhibitors is like you have these checkpoint inhibitors that are normal in your immune system, because the immune system is basically like the immune system we're talking about is cytotoxic, which means cell killer, and you're made of cells.
Yep.
So you need a way to basically put the brakes on the immune system. What happens is that cancers tend to use that to basically survive the immune system. So the idea is, if you can remove the brakes around the cancer, your immune cells are more likely to attack the cancer, which does happen. The other issue is that if you remove the brakes, you might also get some autoimmune related disorders that can be pretty severe. So so it's not yet the magic bullet.
Yeah, as you said, it's cytotoxic. So it's the mechanism for how it kills the cells, can be applied to your healthy cells as well. So as it's killing your cancer cells, it's also doing the same thing to your healthy cells.
It's also doing the same thing to your healthy cells. It works on certain people. Other people it creates the issue. So the idea is to understand why some people develop these immune-related adverse events and not other ones. But in my opinion, if we can get a cancer vaccine, I think that would be a more elegant solution, like way more elegant solution than immune checkpoint inhibitors, and also maybe a more elegant solution than CAR T cells, because CAR T cells are still very hard to make. And they're very expensive, so it's not something that's super easy to deploy. However, if you can get you get a patient, and then you can get a pretty good idea of what the cancer cells are that those specific cancer cells are presenting. They're not present with normal cells. Then you can target these cancer cells more effectively, and probably in a way more cost effective way than CAR T cells in general.
No, yeah. I mean, in the end, the work I'm doing is in the discovery side. So I'm trying to find this antigen, this peptide antigen that's presented specifically in cancer cells. But the application of that can be a lot of things. You can use it to generate a cancer vaccine. So just make the same peptide and just give it to patients and hope that your patient's own T cells starts to learn and become mature and what we call memory T cells that can then target cancer cells. But also, once we have the targets, we have many cool innovations around. I think one of the coolest, most emerging field is designing binders to these peptide MHCs. And people have used many methods, screening methods, like very long experimental methods and automation to try to design antibodies or nanobodies. These are proteins that can go and bind to things. If we find more targets that are shared across many cancers, that gives us opportunity to design more of these binders. And we're now in the age of AI and protein design and computational,
we can talk about
de
that
novo
too,
proteins designed purely, from computational models. And I think it's really cool to see where it's going. I don't know if you've seen, but recently there has already been a few papers showing computational model can design from scratch a totally novel protein not seen in nature that binds very strongly to peptide MHC complex and of a specific peptide, meaning that it can bind to a mutated peptide on a cancer cell.
We have a common friend who works on that.
Yes, exactly. And I actually went to his talk yesterday, so now I'm very good at it, at explaining his work.
We
of course, of course, his paper is out, but he just dropped by in Boston for a talk.
Nice.
Yeah.
Nice. Yeah. So this is a caveat about what I was saying. I was saying cheaper. It's other things. GAA can allow you to make cars. So you can make cars even cars using antibodies. When I said cheaper, more elegant, I was referring to what you said afterwards. It's like the cancer vaccines, like the making, like it's not making sure because it's still biology, but increasing the probability that the cancer doesn't come back, basically, with this type of immunization, and that's way cheaper than having to make a CAR T cell every time. So.
Yes, absolutely. Yeah, I mean that's kind of why the vaccine, the cancer vaccine field has been progressing pretty well, I think. And there's, I think, probably more than 10 different trials ongoing right now, and a couple large-scale trial by Moderna for melanoma patients. The phase 1 showed very good results, long-term immunogenicity and long-term clinical response. And now, we'll see in phase 2, phase 3, what it looks like in more than 1,000 patients. It's all very promising. And as you said, it's very economical because, vaccines are not that hard to make. We've been making them for more, I guess, has it been 100 years? More than 100 years. And so, and there's always room for innovation. And then in the end, when you're, trying to train your own immune system and trying to prime your own immune cells to be able to target cancer cells, it's got a better safety profile, I would say, than putting a whole, new T cells, CAR T cells that's been engineered, or putting a whole new protein like a nanobody, or like some kind of drug conjugate. We'll see. I mean, this is still an active area of research, including the safety profile. But so far, including from our lab, there has not been any serious adverse events from these cancer vaccines, which is very good news.
Yeah, very good news. Worst case scenario does work, which is good, which means you can just try another vaccine.
Yeah. Right.
Yeah.
So far, maybe I might be wrong. So definitely, this is something for people to look up,
exactly.
like in literature. But so far, I don't think we've had fatal cytotoxicity, and we've had that in CAR T cells, unfortunately.
Yes. I mean, it's
since the beginning.
Yeah, it's biology. So nothing is clear cut. There's always but it seems like even in theory, at least in theory, it looks way safer from the theoretical perspective. Because like the worst case it doesn't work is basically the best case for a medication. If you follow the Hippocratic
Right.
the Hippocratic oath, it's like first do no harm. So if you know that you can't actually do harm, it makes it usually a better it's a better way to go about it, which means you can try several ones. You can get even more experimental in the way you design the vaccine, which means you can add more peptides, even if you think that these peptides will not be presented by the cancer cells. In theory, it won't do any harm. In theory, it's still a theory, because you can still, again, in theory, if you include some of these ERV peptides, maybe you have some of your normal cells that also express them for a certain reason, and then you develop some autoimmune disorder. So it's still theoretical, but in theory, it's safer. So it's worth exploring.
It's safer. Yeah, it's priming your own immune system. So your own immune system, I didn't have a chance to get to this, but your own immune system has already had a first screen when you're developing as a baby, called thymic development, in your mother's womb, and then also as a baby. So your T cells are already learned about the things that are presented in your body, and know that they have a certain, what we call central tolerance, so a certain tolerance towards your things that are presented in your cells. So theoretically, as you said, this is a more safer approach than, of course, putting a whole new cell type into your body, like a CAR T.
That's true, but this is again a theory, because people do develop autoimmune diseases.
Yes,
even though they have working timeline. So it's biology. Biology will always be a little bit fuzzy around the edges. Yeah, but without the timers, it is. I mean, it's
yeah.
That
would be a very interesting.
I don't think you can even be born without a thymus.
Right. Yeah.
Yeah, I don't think you would survive your own T cells. So, yeah. So that's super interesting. So, what do you think? Have you done any single cell work recently?
Single cell, interesting. I haven't really. It's been something very it does feel like a distant life, but I think it's something that's definitely I'm definitely thinking of getting back into. I think it's still a very cool technology. There's a lot that you can do with single cell, and I think a lot of single cell work requires going into the data set, annotating things, trying to make sense of it. And of course, the expertise is very important. But I see a lot of potential in how language models potentially can help accelerate like how we understand large, such multi-omic, multimodal single cell data sets. So that's actually something I maybe not do myself, but I'm looking forward to see where it goes. Like, can people take old data sets and learn new things? Because we are able to like combine many different fields of expertise through the large language model. We're able to integrate many subfields, looking at rare cell types. Can we do something and learn something from the many data sets that we have? Probably like billions of cells now that we've generated over the past 10 years, I think.
Yes, definitely. I'm not sure the large language model architecture is the best one, but there's definitely like I'm 100% confident that we can learn a lot from all the data sets that are freely available. Let's say the billions of cells all over the place. It's interesting working on single cell and large language models is it's interesting because large language models do have quirks, but I think we don't talk about enough. And one of the big quirks that everybody notices now is the M dash. They tend to write with M dashes, and it's to me it's that M dashes has been it's fascinating to me. Like this M dash, I've been thinking about this M dash like almost every day.
Really?
Since the first time. Yeah,
Okay.
since the first
Tell
time I
me
saw
more.
it.
Actually, yeah, I'm so curious.
Because it's if it were like if it were something common in the way people write, that would be something expected. But it's the M dash is so uncommon that most people don't even know how to find it on the keyboard. So why do you if you have a model that's basically outputting it's still simplification because there's stuff around that basic concept, but the basic concept is that you're outputting the next token. So there's the probability of the next token. The probability of having an M dash should be like super low. It should be so low that you barely see any M dash, but you see a lot of them. It's yeah, and this is weird, and it means that if we apply the same type of models to biology, we will get M dashes.
That's true. I mean, it's such a black box how these models are trained. I mean, perhaps M dashes are very common in old books, and it's trained on PDF files of archives of millions and millions of books. And yeah, it's a black box.
It doesn't have to be. I think it doesn't.
I know
have to
Reddit
be.
doesn't seem to use M dashes, and it's trained on Reddit posts. But who knows what else it's trained on?
It's trained on Wikipedia. Wikipedia doesn't have M dashes. It also have these quirks of writing, like using. It's also using terms that are not very much present in old books. At least in my experience, they seem to be like highly like because just people started writing, especially like more and more in the past decades, like using more superlative language and using words like delve into, and nobody writes that anymore. Like it's super rare to use.
Right. Yeah.
But they use that, and they have their own ways of talking that doesn't follow the distribution of human the way humans use English, and that's what I've been thinking about. I'm not joking. I've been thinking about M dashes for almost every day. It's true. Looking to understand
Yeah.
why do you think, where does this come from?
It's
Because
crazy. Yeah.
yeah, it's crazy because this is a very, very strong bias. Like if I don't like the term hallucination
Mm-hmm.
because I don't think they hallucinate. I think they do something very different from. They don't hallucinate. They don't lie. I mean, these are human concepts. They do something very different. But if the if I were to call something hallucination, that would be the M dash.
Okay.
And to me, when I see something like that, I'm like, maybe we should not apply that so readily to biology because it's something we don't know. We're looking for something we don't know. We know that there's things that we don't know that we're looking for, and then we're processing
Mm-hmm.
that through large language models that we know for a fact has some quirks when it works on natural language processing. What what hallucinations will it impart on the biological data that it will output?
Mm-hmm.
And yeah, so that's why I've been thinking about that, and I'm not 100% certain that using these things without having a clear idea of how they basically just how they work
Mm-hmm.
internally, like a little bit understanding the M dash thing. Without understanding the M dash, I'm not sure that this would be the best way to go about it. I would yeah. I think yeah, that's it. I think we should develop like simple architectures that can probably do the same thing.
Mm-hmm.
And
Yeah.
yeah, and mhm.
No, that's definitely true. Let's switch role because it's very interesting. I do want to know more. So how would you envision a way to build a model that's able to help us interpret, these single cell data sets or even like, biological findings? Because I think in the end, what I think is that we do have millions of research papers, and not everyone is able to be an expert in every field. And of course we should be talking to each other, but is there a way to make this ideation process easier so that you can find a person to talk to that's an actual expert, but like you wouldn't even get to knowing who to talk to if you haven't found some tiny quirks, and could a model that's trained maybe in a better way on all these papers and data sets, and can be integrated and help us
Mhm.
with that?
Yeah. For papers, I think LLMs are fine because it's natural language. It's easy to spot things, uh, and uh, and also like these LLMs are not just LLMs. They used to be just LLMs, and they were the first ones were put into production as LLMs, and this is why that's where the whole assignation things comes from. But then now it's not just that. There's a lot of things on top of the LLM. And there's also some there's quite a big deal now of reinforcement learning above the LLM, and that reinforcement learning is basically having a human in the loop. So you have people scoring the answers and seeing, yeah, and we can't really do that with biology. We don't have enough people, and especially we don't know what we're looking for. That's the big difference. It's like if you use, for example, an LLM um, to help you to do literature review. It works really well, especially if it gives you the references. If it doesn't give you the references, it doesn't. I wouldn't use it.
Uh-huh.
But if you use an LLM that gives you the references, it's pretty easy to check because you can go to the reference, you can read the English, you can understand basically what's happening. Um, you can do it yourself, and you can assess if this is a good answer or a bad answer. If it gives you a biological answer if it gives you gene expression or protein expression or a protein sequence, how would you
Uh-huh.
evaluate that?
Uh-huh.
And that's the big issue. And if you want to have if you want to create an LLM and then have a reinforcement learning above the LLM, how many people will be able to assess the quality of the gene expression of the LLM. Like very, very few people, if any, because if it's like ongoing research, the number of people could be exactly zero. And so that's why I think I'm not really sure that these are the right models, because we know for a fact that when you use them on natural language, they don't work that well unless we have these other types of algorithms on top of them. And I don't see how we can apply these algorithms to biology. And even when we apply these algorithms, we still get the M dashes.
The cursed M dash.
Cursed M dash.
Yeah.
Yes, but I still think that AI has a lot of things to bring to the table. I just don't think the right form is the current form of transformers. That's the caveat. We probably need a different approach than just stacking transformers over transformers over transformers. I think that would be that's my take on it. It's like we probably need to develop a new types of model that we can better control when it comes to just not really control. It's not the right term. Like more interpretable models that gives you more interpretability, that less of a black box.
Mm-hmm. Yes.
Yeah.
No, that's fascinating, and yeah, I think it's something that's definitely worth pursuing.
I think so.
It's just for the how interesting it could become in terms of getting this.
I think yeah, definitely. Protein structure is pretty cool. Like for protein structure, there's a lot of things happening.
Yeah, so I mean, this is definitely an area I want to get into. So what we've talked about is mostly things I did in my PhD, and that I mean that was only a month ago. So
Congratulations!
It's something I just did.
Yeah, I'm so
but
very
yeah,
happy
I
for you.
thank you so much. Sorry, this is it's been a long journey, and yeah, lots of sweat and tears. But yeah, I mean, now looking forward to the future, I think the world of protein design and protein modeling it's also very fascinating. There are really some really cool things I think you can do. The problem in our field that I guess what our field would know the best actually is that people in the protein design field, they don't really know very well, or it's on the back of their mind, but they never really think about it. Is that once they design all these proteins and they want to put it into people, these are very immunogenic, actually. These proteins are not made for people, like we talked for the past hour about how we could find peptides from cancer cells that our cell can recognize, and we can guide it to kill the cancer cells. The proteins that people are designing computationally, those are like they're all the ways that are very different from our normal proteins, and they're definitely going to be very immunogenic, meaning they go into your body and like rapidly, instantaneously, all your immune cells are going to see it as something foreign, something as much as like a virus and a bacteria, even more so possibly. We don't know yet because we have not put a de novo design, computational design protein into a patient, into a human. But because they're designed based on these models, they're not similar in sequence similarity to humans. They're possibly further away from us than bacteria and viruses are from us.
Yeah.
And of course, there are people who are thinking about this, but I think it's not on the forefront of the field at all. You don't see it when you read articles about protein designs. You see about all the amazing things it could do, and it can do all these things that they could have customized it to do. release certain peptides at a specific time, or like be sensitive to pH, or be able to, fluoresce different, different colors, different fluorescence, have, very interesting pore shape structures, so like nanoparticle structures. But immunogenicity is going to be a huge issue in this
definitely. Definitely. It seems to be such a hard problem. It's like, how would you go about that?
It's tough. I mean, people are doing it. And an example I saw from a talk is that you could, and I mean, this is all very early kind of work, but you could think about it as, and perhaps you should invite Bing Shu, who this is the talk that I saw
Oh, he's already
onto your
invited.
podcast. Yeah. Oh, great. Yeah. No, that's awesome. He can tell you way more about this because he's the one leading this project. But essentially, you have prediction models that can predict binding of these peptide to the MHC molecules, and of course, you're very much involved in a lot of the early prediction model work. But yeah, I mean, if you integrate this model, the idea is that if you integrate this model into these computational protein design model, you can try to remove certain epitopes or remove certain characteristics of these peptides that would bind, so that you create a protein that hopefully retains the function you want it to be, but whose sequence of amino acid does not combine to create something that could be presented on the MHC molecule. And how well that model would work, whether it could still create, these really beautiful de novo design proteins, I think we still don't know, but that's what someone is working on for sure.
Yes. But it's but even if you have a model, it's not 100% guarantee that you won't get the some theory. Yeah.
The limitation is also in the prediction model for presentation.
Exactly. Yeah. Because it's super far. It's alien proteins. They don't look like. They might not look like anything else in nature. Yeah. Yeah. But they could do. But maybe if you could encapsulate them, you could create like this. Like I'm thinking like this is more like science fiction thing, but maybe you can have like small machines inside. Like you create a lipid. A lipid membrane. When you have this
delivery would be very important. Exactly. Yeah.
And you package it with some form of proteasome that would like just cut them when the work is done. I don't know, but it's you're absolutely right pointing that out. It's true, and it's a hard problem. It's a hard problem. But the problem of designing the proteins is it's such an ongoing thing now that it's we have to figure that out first, and then we can figure out the how to make them less immunogenic and more usable in a clinical context eventually.
Yeah, but all exciting work. Definitely things that can keep us excited and happy to go into lab and try out experiments.
That's true. So what are your plans now that you're a Dr. Manakongtreecheep? So what's happening next?
Yeah, I mean, I'm exploring. We have some early stages projects now, thinking about exactly what we've been talking about, the next step, like thinking about protein design binders. We're also thinking of experimental screening platform to match the T cells. We talk a lot about how T cells would be able to be trained by the vaccine to target specific peptide on the MHC molecules. Can we find the identity of those T cells at the same time as the identity of the peptide it's targeting? Can we, make predictions when we take our blood out and you get these, T cells that we sequence? Can we make a prediction for which one can actually bind to a specific target that we're looking for? So all these, it's a very much hybrid experimental and computational project. And so it's early stages, but this is the kind of area where I want to get into. So like now that we did a lot to hopefully advance the field of antigen discovery, finding more targets. Once we find the targets, can we utilize it? Can we find binders to it? Can we predict T cells that would bind to it so that we can target those T cells specifically? This is the kind of stuff I'm interested in.
So, you're getting back to single cell.
But yeah, I forgot to mention, that's right. Like, I mean, to get to this type of screening technology, you do need the ability to, identify single cell, which T cell is matched with what.
Yes.
So yes,
Yeah. So
single cell will be once again what
it's
I'm
exactly
going
what
into.
I did. So funny.
Yeah.
MHC, and then you need the other. You need to understand the other side. So you go.
Exactly.
Yeah.
Yeah.
You get to this T cell side. Yeah. So TCRs.
Mm-hmm. T cell receptors. Mm-hmm.
Mm-hmm.
Yeah.
Yeah.
Predicting T cell receptor sequences.
Mm-hmm.
For sure.
Yeah. So are you thinking more predicting, like finding the right TCRs that already exist, or creating new TCRs, like for example CAR T cells or maybe nanobodies? It's what would you? Yeah.
I think we need to do both.
Okay.
We need to figure out how to do both. I would say there are gonna be cases where it's better to know which T cell in our body is already very useful and then expand them. And there are cases where we need to design binders or put in the right T cell receptors to make those T cells target cancer. So yeah, I mean it could go both ways. The first step is to find that target, which I hope we will continue to work on this mass spectrometry Pepyrus technology and be able to find more targets. And from there, we can start to think about, on the T cell side. So yeah, I mean, a lot of exciting things. And I mean, you mentioned the prediction model. I think it's something that definitely can be improved. The prediction model for presentation, for the peptide presentation. This is especially true for many patient population that may not be, they may not have common MHC genes. So these MHC, there's so many of them. And, as people migrate out of Africa, we evolve and these MHC gene changes over time. And, Europeans have certain MHC genes that present certain types of peptides. And then Asians or North Africans who have a totally different set. And not all the prediction models are not good for every actually genes or alleles. So I think that's definitely another thing I would love to put my just to get some feel on and explore, because in the end I think it's a really cool therapeutic modality, this cancer vaccine or like targeting peptide MHC on cancer cells. And I think looking ahead in the next 10 years, 20 years, I think other countries are going to want to try these trials. And it's already started, actually. There's some trials in China, some trials in Australia. I don't know about Morocco, if there's any cancer vaccine trials.
No, I don't think so.
Perhaps not.
Yeah,
but I think eventually, once you're able to scale the manufacturing of these vaccines, and once you find more shared targets that are shared across cancer, it's easy to manufacture. You can make a lot of it just for you can make just one, but make a lot of it, and you can give it to like 100 patients. That would be the dream. Or 1,000 patients. That would be even more amazing. But yeah, I mean, these are things that I think the world would start to come to. Of course, if the trials all work out well, we're all waiting for results from the trials. Gotta make sure we have like good empirical evaluation, good statistics, good number of patients, good evaluation and monitoring of side effects and all that. But hopefully it leads to more adoption and more use.
And it's it's good that you talk about the differences, the different alleles, because it's true that the data sets that we have are usually not like they're biased towards Caucasian population because it's just like where first data start to get to be gathered. So we have a lot of, for example, HLA, like the European HLA is like HLA, it's the MHC for human beings, are vast majority of what we have, and but they're also the most polymorphic genes in humans. So there's so many different alleles, many different
more than
alleles all over the
10,000.
place. Yes, it's huge. It's huge, and and you have places like Africa that are the problem we have in Africa is that we still don't have enough data. It's super genetically diverse, where we don't have enough genetic information on the population, and which means it's actually hindering this type of the application of technologies, because it's not, because you can find a peptide that's being presented by an European allele, that this peptide will also be presented by an African allele. So you can find the antigen, the TAA, but might be completely useless in other parts of the planet. We need to get to work. We're actually doing it.
Yeah.
We're actually getting data in Africa just because we need that to be able to push forward with this type of technologies, and therapies.
That's really awesome. I didn't know that you're involved in that.
Many different aspects. There's more and more labs in Africa that are starting to be really well equipped. So UM6P, my university, is pretty nice. But I also have collaborators also in Ghana and different places, and we starting to get data. We have exome-seq, we have RNA-seq. We're starting to get data on the African populations, and also on the African cancers also. Because they might be different. They also they might be different, and we're starting to map out this huge diversity that we have in Africa. There's a paper that was published this year in the communications biology it's not my university, but it comes from Morocco, and it's the first phase of the Moroccan genome. So
wow, that's so cool. Yeah.
It's cool. It's cool. You can read it if you want to. It's pretty interesting to see the different different haplotypes, basically, from the maternal side and the paternal side. See where Moroccans come from.
Wow, that's awesome. Yeah. Wow, maybe also we could, if you're doing a lot of these African population, perhaps if you do any mass spectrometry, we could
we'll have
work
to
together.
do it at a certain point.
Yeah.
We have a mass spec. We have a mass spec, and the goal is also to do mass spec. I mean, as much sequencing as we can, plus what I'm more closely involved with, which is more the AI side and the methods development.
So cool, Tariq. This is awesome, and perhaps we should find a way to collaborate.
I think we should do that. Yeah, we've been talking about it for a long time.
We all have the same end goals.
We have the same trajectory. You're doing this.
Yeah, right. That's so true.
Yeah. We should get something started. Yeah, we should have a meeting and then get something started. That would be awesome.
100%.
Really awesome.
Well,
yeah, I know I have an experiment to get to.
Yeah. This was great. This was so fun. Thank you so much.
Thank you, Cass.
I learned a lot from you, and also it was this was a really fun conversation.
Yeah, and I really enjoyed reading and learning about Pepyrus. It's I really like it. Like, honestly, I read it, I was like, oh, this is really cool. It's just a nice idea.
We'll see where it goes. Yeah. So nice to see you. Thank you, Tariq.
Nice to see you.
Bye-bye.
Bye-bye.